start immediately with the first speaker. It's Ingrid Fernandez Santana Martins from UNESP in Brazil. And she will talk to us about mesdoperin-like antimicrobial peptides.
Thank you. Sharing here. Yes. So, good morning, everyone. I'd like to present to you my study that, as Kirk said, and this study has the supervision of my advisor, Professor Alexandre, and also has an experimental part that was made by the lab of Professor João Rougier. And I'll start with the experimental motivation that got us into this project, that it is the antimicrobial peptides are mostly positive and they have strong interaction with the bacterial membrane that is mostly negative. Professor João lab worked with two different antimicrobial peptides, MP1 and HMP1. On the next slide, I'll show you the difference between them. That they did some experimental about the vesicle permeability, a vesicle made with a mimetic bacterial bilayer. And they show different leakage from the vesicle and different pHs. The MP1 is most effective on the pH 7.4, but it is effective at all three. And the HMP1, it's not effective at pH 6.5 and 7.4, but it is at pH 5.5. And this is why the MP1 peptide is obtained for a wasp called Polybia paulista, and it has 3 lysines. And the HMP1 has histidines instead of lysines. So at pH above 6, it is deprotonated. The histidines are deprotonated, so the peptide gets a negative charge, so it would not like the bacteria bilayer. But at low pH, it would be positive, so it will be attracted to the bilayer. I built the system with 120 lipid molecules at the ratio of 7:30, POPC, POPG. The POPC is a zwitterionic lipid, and POPG is a negative one. I did the simulation in the same salt concentration and in the same temperature as the experimental. And I did a lot of replicas with different initial velocities at the 3 pHs. It's important to notice that the protonation state of each amino acid I choose based on its pKa and the pH. And the lysine has a pKa of 10, so it would be mostly positive. And the histidine has a pH of a pK, at 6, so it will be protonated only in the 5.5 pH. In this presentation, I'll only show you the best simulations that I got from all these replicas that are MP1 at 7.4 of pH and HMP1 at 5.5. I also built the system with the peptide on a alpha helix configuration because we have experimental evidence that showed that this is the predominant configuration when the peptide is interacting. I used NEMT and the charm force field for this simulation. The objective of my work is to understand how the adsorption happens, because we can have different types of adsorption, and which amino acids are interacting the most, are most important on this adsorption. And what happens with the lipid bilayer when the peptides interact. So the first result that I'm going to show you guys is the MP1 at pH 7.4. This lipid bilayer, when it is equilibrated and everything, has a thickness around 40, 42, something like that. So I measured the distance of 3 parts of the peptide that are the center of mass, the N terminus, and the C terminus to the center of mass of the bilayer. So in this first plot, when the distance is below 20, it means that it is inside the bilayer. When it's above, it's outside. You can see in this snapshot that the peptide at the end of the simulation gets all inserted on the bilayer. We have another experimental result by fluorescence that show that the tryptophan tends to stay inserted in this interface between hydrophilic and hydrophobic part of the lipid bilayer that is around 16 angstrom distance from the center of mass, 16 to 17 angstrom. And I did a histogram of this distance between the tryptophan and the center of mass of the bilayer. And the most frequent distance on my simulation is 16 angstrom distance. That is the result that we expected that the experimentals see. I also want to show you my density profile for this simulation that I got the MP1 outside the bilayer in the beginning of the simulation, and it gets all inserted on the bilayer as the simulation occurs and the adsorption occurs. I did this heat map of the lipid bilayer before the peptides adsorbing and after. The first one I got just in the beginning of the simulation, and you can see a difference between the narrower part and the most thick part, that is 36, 37 angstrom to 42. And after the adsorption, the narrower part of the lipid bilayer, that is where the peptide is interacting, gets to 27, almost 28 angstrom. So it's a difference around 9 angstrom. That it's the peptide is inducing a narrowing in the lipid bilayer. Also, I wanted to see what happens to the secondary structure for the MP1, and almost all amino acids stay in alpha helix configuration during the entire simulation. And just the C-terminus part gets unfolded. I also wanted to see the difference of energy between van der Waals and electrostatic interaction. And as a result, I got obviously the electrostatic part, it's way higher, way more negative than the van der Waals part. And the peptides that are interacting the most are the N-terminus, that is positive because of the pKa of N-terminus and the pH of the simulation, and the N-terminus part of this lysine 4. The lysine 5 and the lysine 11 that's closer to the C-terminus is not interacting that much than the N-terminus part. And the van der Waals energy, the most interacting peptide, it's the tryptophan. I did a simulation for HMP1 at low pH that has the same charge that the MP1 is. In this pH, the HMP1 has the same charge, that is 2. It's kind of hard to see, but my initial distance here, it's around 30. And then rapidly the N-terminus part gets below 20. And the rest of the peptide, it's not getting inserted on the bilayer. You can see in the snapshot that the tryptophan, that is this orange one, it's in the interface. That's more or less the same position that it got on the MP1, but the rest of the peptide, it's outside the bilayer. And the distance between the tryptophan and the residue, the center of mass of the bilayer, it's interestingly the same that we got from MP1. That's around 16, a bit higher, like 17. That's the result that we expected from the fluorescence experiment. So I got the same most position that I got the tryptophan in the simulation. And then I also have the density profile for the simulation, but we see that HMP1 does not get all inserted on the bilayer as the MP1 did, because it adsorbs on a vertical way, not a horizontal way. And also, I did a heat map for the thickness of the lipid bilayer, and before it varies from 30, almost 31 to 44, And after the adsorption, I have 22 to 44. That it's around 9 angstrom. Again, difference between before and after the bilayer, the peptide adsorption in the bilayer. And the secondary structure, it's not that stable like MP1. It gets unfolded a bit more, but it also has a helix configuration here. And the electrostatic energy show almost the same behavior as the MP1, but this C-terminus part, it's not interaction that much, but there's a matter of distance because this part of the peptide does not get adsorption in the bilayer as MP1 did. So we can conclude from this simulation that N-terminus is leading this adsorption because also if you see the video of the simulation, it starts at N-terminus. The adsorption, the interaction starts at N-terminus. We can see some bilayer modifications when the peptide is adsorption. The MP1 has more stable structure. This needs further analyzing. And I have some perspective that I'm still working on. I did some constant pH simulation with the method by Radak, published in 2017. And I have also extended the simulations to see if the HMP1, in some time, it gets adsorted, just like MP1. I don't know if this is what happens or it's a matter of time. So I extended the simulation. And I would like to thank Professor Alexandre, that's my advisor, Professor João and Thaisa, who are the ones that made the experimental part of this work, and all you guys for your attention.
Thank you. Great for a beautiful talk. I'm sure there are questions, so we will encourage again students to ask questions now, either by raising your hand or in the chat. So we have already a question in the chat from Basilio, who asks how you implemented the difference in pH in your simulations, and he apologizes for not arriving on time.
No, the pH, the protonation state that I chose was based on the pKa of the amino acid and the pH of the simulation. So if the pH of the simulation is, let's say the pKa of the lysine, it's 10, and the pH of my simulation 7.4. So it would be protonated. And the same for the histidine, it's around 6. So the simulations that are 6.5 and 7.4 is deprotonated, and in 5.5, it's protonated. But I'm also doing the constant pH simulation to see if this pKa, it's varying when the peptide is interacting and when it's not.
Okay, then we have another question from Clarice, who asks, did any controls with just the plain membrane or just the protein to compare the structures of both without the presence of the other?
Yes, I did a simulation with just the bilayer. I equilibrated the bilayer before I could put the peptide inside the simulation box. So the bilayer is already equilibrated. And then I have this peptide simulation just in water, so I can compare the secondary structure and other things. But I did not show it here because it's a lot to show, but I did.
Thanks. Any other questions?
Just a short question. There's no more student or postdoc asking questions?
Not at the moment.
Do you always see the same pathway for the peptide to insert on the membrane? I know you have this favorite path part by the N-terminal, but during the simulation, have you seen trying to get parallel first or stuff like that? What have you observed?
Well, to see, because I needed to see a lot of ways, so I started with a lot of replicas in different initial configuration, and I saw that the ones that approximated the bilayer on a horizontal way did not got all adsorbed like this one. So we just get closed and stay like that. the ones that start with the N terminals gets inside, are the ones that are able to get inside. And we also have some experimental data that show this. But
we have time for one more question.
Yeah, can I ask a quick question? This is very nice work. Have you ever considered hooking on to your lipids, glycolipids? Because in many bacteria, the surface, is not just the lipids that are there.
mm-hmm.
Can have glycolipids, and it would be interesting how they interact with these microbial agents or antimicrobial agents.
Yeah, this is a great question. Actually, I do not have some control in this free simulation of where the peptide will get first. So we could get near the glycolipid, near the POPC, near the POPG. I do not have this control. So I would need to get a lot of data, generate a lot of data, so I could cover all the possibilities. So for this first thing, I preferred a simpler bilayer because of this statistical problem. But it's a good idea. I could definitely consider trying it.
Yeah, I think you'd be unique in doing one of the first MD simulations on that. And it's of great interest.
Okay, that's great.
Mm-hmm.
It's a great idea.
Oh.
Thank you.
Yes, I'm still in Brazil. I'll be back in a couple of weeks. I call you when I'm back. I noticed.
Okay, so I think it might have been a sign that we on time for the next talk. So let's move to Madeline Galbraith from Rice University in Houston, who will now talk about stem cell differentiation. Yeah, perfect. Go ahead.
So thank you all for giving me this opportunity to talk today. I'll be talking about stem cell differentiation by random parametric perturbations. So stem cells, also known as embryonic stem cells, Waddington was one of the first to kind of give an abstract idea that if you think of these stem cells as a ball rolling down this landscape of hills and valleys, the hills and valleys are these constraints on the system that lead to some sort of final state. And so you can think of that as the embryonic stem cell going through this landscape to end up as other cells within our body. And now, quite a few years later, we understand that this differentiation is controlled by a lot of different transcription factors, one of which is NANOG, and that dysregulation of the differentiation can result in diseases like leukemia. And so one of the ways you can understand embryonic stem cells is to understand the gene regulatory network. And so the stemness gene regulatory network, it is the one which governs differentiation of stem cells. And as we know, many different gene regulatory networks play crucial roles in many different processes, and we're just focusing on the stemness one today. And so you can quantify these gene regulatory networks using many different approaches. There are stochastic simulations, many-body problems, Langevin equations. You can even just use an ordinary differential equation. But there are shortcomings, one of which includes the fact that typically a lot of these other methods, you end up simulating just a standalone circuit, and you can't get information about like the microenvironment within the cell inside of these circuits. And they also depend on kinetic parameters, which are derived from experiment, which I don't know, they tend to be very difficult to get from an experiment, especially some of the newer circuits and networks. And so one of the methods that was developed to overcome this was RACIPE, which is random circuit perturbation. And so this is based upon the idea that instead of you needing to know every single gene within a network. You can just know the core gene circuit, and from there you can develop a range of parameters. And if you perturb those parameters, that reflects the differences in the microenvironment within a cell. And this model can be used to identify robust gene states, understanding network regulation, and even understanding which parts of the network are the more important parts. And so on the left side here is a depiction of how RACIPE works. So you start with your mathematical model in which you have some sort of production rate G multiplied by your transcriptional regulation, which we use a shifted hill function. And so if you look on the bottom right, you can see that for activation, the hill function goes up, and then also for inhibition, it's the opposite. And you can see how the different parameters, the threshold of regulation, the cooperativity N, and then the fold change, lambda, all affect the activation inhibition regulation. And so that is added for every regulatory link. And then you also have your degradation rate of the gene itself. And so RACIPE, you define ranges for these kinetic parameters based upon your system, and then you place them into your mathematical model using random initial conditions. You solve those deterministically, you get your states, and you repeat for however many kinetic parameter sets and initial conditions you want. And then at the end, you have many different solutions to your states. And so if we look at a little closer in detail, so RACIPE has two major assumptions, one of which is that all genes have the same maximum production rate, as this is determined by the transcriptional rate. And then all the regulatory links, which we define using the hill functions, are equally likely to be either functioning or non-functioning. And so what that means is if you have some sort of isolated gene, say A, if you apply the model where you have just production and degradation of A, you would have the expression of the gene come out to look like this. And then the median level would be the threshold of regulation, where you can see that about half of the gene expression is below the threshold and half is above the threshold. And so when you apply that to a larger circuit, you look at your circuit, you look at the gene, and only take into account the inward regulatory links for that gene to determine its threshold value. And so for the inward links, you use the standalone distribution for those genes, and you model this, the gene expression for A, using only the inward links to obtain an approximate threshold for that gene. And so if we're going back to our model for stemness, you typically have some sort of graphical representation like this, where you have many different genes together. So in the stemness circuit, you have 8 transcription factors and then 1 protein-bound complex. So the protein-bound complex is OCT4 SOX2, and the red links in this network indicate activation. For instance, the activation of GCNF by GATA6. The blue links indicate inhibition, such as the inhibition of NANOG by GATA6. And then the purple links indicate the binding of SOX2 and OCT4 to the dimer, and the reverse is indicated by the brown arrows. And so the mathematical description for SOX2 then looks like this, where you have the production rate, multiplied by the shifted Hill function, which is an activation of the protein-bound complex onto SOX2, followed by the degradation of SOX2, and then also the binding of the monomers to the complex, and then the unbinding of the dimer. And so from here, we can use RACIPE on this model to get the steady states and then look at them. And one of the first things you can do is you can do a clustering analysis. So on the left, you have the principal component analysis of the steady states, and you can see that there are two large clusters. So this is the probability density projected onto the first and second principal components. And so this tells us that we have two somewhat distinct clusters in which the gene states live. And so on the right, you have the hierarchical clustering analysis, in which you cluster all of the steady state solutions based on their approximate gene state. And so you have around 15 gene states. And so how these two compare together is you can actually project the actual gene states onto the principal component analysis, and there's a fairly even split between blue in the first 3 columns, followed by red in the last 6, compared to red in the first 3 and blue in the last 6, which somewhat correspond to the actual projection onto the principal components. And so we can even compare this to experiment. And so you see that you have around 4 of the predicted clusters from RACIPE actually match those within the experimental results. And so we can also look at how the regulatory links behave. And so one way to do that is using the Kolbeck-Leibler divergence, in which the higher the value of the divergence means the more important that link or gene is within this network. And so these two charts on the left are described in more detail in this network, where the relative size of the node indicates its relative importance, and then the thickness of the links between the nodes explain their importance. And so you see that SOX2, the protein-bound complex, OCT4, and then CDX2 are all very important within this network. And so in conclusion, RACIPE can identify many different clusters and gene states, and even predict experimental states. And from these results, we can say that CDX2, OCT4, SOX2, and the complex form a very important subnetwork. And so with that, I'd like to acknowledge all of the help from Donia, Mingyang, and Ben, who have helped with this project, and then our advisors, Herbie Levine and Jose Onushik. So, thank you, and I'll take any questions.
Thank you very much. I'm sure there are questions. Let's start with students again. Either raise your hand or just talk or write a question in the chat.
Yeah, so sorry, I was just trying to formulate my question. That was beautiful, Madeline. Thank you for sharing it. So how hard would it be to incorporate something like external cellular conditions or even like metabolite conditions in the cell into this model to make some predictions as well?
So if we're talking about just like the cellular environment in itself, we take that into account with the different perturbation to the parameters. So we don't set a specific parameter, we just set a range and model within that range. You can also expand this so that it's a fairly new project, and so we have expanded it somewhat. So you would just have to add in the actual like mathematical term for that model for like a metabolite, if that's what you're asking.
Yeah, that's exactly what I'm asking. All right.
Yeah, I
Cool.
think it'd be fairly easy.
Thank you.
Okay, so we have another question from Alma in the chat, and the question is, how much effect does the stochastic noise have on the cell state transitions versus the deterministic internal or external cues?
So we don't actually have any stochastic noise in this version of RACIPE. So we just simulate within a range of parameters. But that range, within that range, you end up with just a few different steady states.
Thank you. Any additional questions?
Yeah, I have a quick question. Since you were able to look at the stem cell differentiation, are you trying to have the stem cell differentiate into a particular type of cell, like a brain cell, a heart cell? Is that the long range goal?
So I don't actually know if that's the long-term goal. So this was just like one of the networks we could look at. And so you can apply this to many different ones. The stem cells we looked at were just general. I don't think they were any specific type.
It's just the initial development stage.
Okay, that's very beautiful work.
And we have time for one more question. Real quick. Okay, so I think then we thank Madeline again for a nice talk and answering the questions. And we move to the next talk by Feng Wen from UT Austin, who will talk about membrane bending of by protein phase separation. Yeah, it's your stage.
Oh, can you hear me?
Yes, we hear you now.
Okay. Okay. Good morning, everyone. I'm Feng, and today I'm going to tell you about some work I have been doing to understand how protein phase separation can bend membranes. Membrane bending is found all over the cell. Trafficking vesicles are one of the most obvious examples. They are constantly being formed to carry lipids and proteins from one place to another. You can see they are highly curved. So what kinds of protein drive this curvature? Well, most of them have one of these two features, a curved scaffolding surface or a wedge-like insertion. Both of these ideas require specific structural motifs. However, it is interesting to note that these proteins also have large intrinsically disordered regions. Here, I have the same two proteins, epsin and amphiphysin from the previous slide, but now I'm showing you the full length of the protein, which has large intrinsically disordered regions. While we know a lot about the structured region, we don't know much about the disordered regions. One of the most interesting recently discovered behavior of IDPs is that many of them participate in liquid-liquid phase separation. And phase separation can not only occur in solution, but also on 2D membrane surface at a lower concentration. This brings us to think about a question: how will assembly of disordered protein on membrane surface impact membrane curvature? We don't know yet whether the actual proteins involved in endocytosis participate in LLPS. Several labs are working on that. So meanwhile, we ask this question more generally using a model protein that is known to undergo LLPS. We choose fast LC domain because it has been so well studied as a LLPS protein. Another important property of fast LC is that increasing salt concentration drives LLPS. So we made giant vesicles and tethered them to cover slips and added fast LC. The fast LC has a histidine tag that bound to nickel-NTA lipids in the membrane. Right away after adding the protein, we could see protein binding and phase separation on the 2D membrane surfaces. But before long, many of the phase separated region began to deform, and we saw what appeared to be lipid tubules lined with protein going inward into the GUVs. Besides, some of the tubules had an undulating appearance, while others showed a structure like a string of pearls. Still, there were some tubules that were so slender that their morphology could not be precisely determined. Moreover, tubules were observed more frequently as the concentration of FAS increased. At 500 nanomolar, most of the vesicles didn't have tubules, but by 1 micromolar, almost half had tubules. There was a plateau in the number of tubules, likely owing to the appearance of three-dimensional protein droplets in the surrounding solutions. Most likely, there is a computation between the membrane surface and the droplet for the protein, and that limits the number of tubules. Next, we varied the protein membrane and protein-protein interactions, which also influence LLPS on membrane. We did that by varying the nickel-NTA fraction and salt concentration. We took images under different conditions and calculated the phase separation event fraction and GUV tubulation fractions under each condition. The results demonstrate that the formation of protein-lined lipid tubules is strongly correlated with phase separation of fast LC on membrane surface. So why does LLPS on GUV membrane cause inward tubules? The morphology of the inward unduloid tubule suggests that LLPS reduces the area of the outer leaflet relative to that of the inner leaflet. So we might expect such a reduction in area when LLPS pulls the lipid head groups together, creating a compressive force in the outer membrane leaflet that increases the local density of lipids. To examine this idea, we developed a continuum mechanical model in the presence of LLPS, in which a compressive stress is applied to the to one leaflet of the lipid bilayer. The model is based on a Helfrich framework, and the simulation result shows the tubule shape transition with different spontaneous curvature and rigidity. And from the simulation of the model, we can see the tubule diameter increase as the square root of the membrane bending rigidity, and the tubule tends to become pearl-like structure when LLPS becomes more rigid. To examine the predictions, we measured the diameter of lipid tubules as a function of membrane bending rigidity through variations in membrane lipid composition. As we expected, we observed a substantial increase in tubule diameter, and the data were reasonably well fitted by a curve in which tubule diameter was proportional to the square root of bending rigidity. Furthermore, as the protein layer become more rigid at high salt concentration, we observed an increase in tubule diameter, and the incidence of pearl tubule as salt concentration increased from 50 to 250 millimolar, which is also in agreement with the simulation. Furthermore, we wonder if this mechanism is significant enough to bend a cellular membrane. So we expressed GFP nanobody on RPE cell membrane and extract GPMVs. We can see GPMVs look a lot like GUVs, and we added a GFP. We added a GFP to the N terminal of FastLC to achieve protein binding. So when GFP alone binds, no tubule was seen in GPMVs. But when we added GFP fast LC, we see membrane indentations and tubules. The tubules again had pearled shape. The tubule diameter ranged broadly from 150 nanometer to more than 1 micron, which encompasses the scale of many cellular structures, including filopodia, dendritic spine, phagocytes, and many organelles. So in conclusion, we demonstrate that liquid-liquid phase separation on membrane can generate considerable compressive stress that drives inward tubules of physiologically relevant dimensions. The ability of this mechanism may be relevant to membrane bending by protein networks that drives outward protrusions from cellular surface, such as filopodia, dendritic spine, and verubuds. This requires further research. That's all. Thanks for your attention.
Thank you very much, Feng. We already have a first question from Mauro, who is asking if you have any idea what is happening at the molecular level when the rigidity is changed.
Oh, sorry. Could you say that?
So the question is if you have any idea what exactly is happening at the molecular level when you change the rigidity?
At the molecular, I don't quite understand the question. So,
yeah. So the question is what if the molecule, what is happening on the molecular level when the rigidity is changed? But maybe Mo wants to clarify the question.
Yes, can you clarify this question? So okay. So when the salt concentration increase, the interactions between so there is like a multivalent interaction between the phase separation proteins. So when the like in my circumstance, the salt concentration changed, the multivalent interaction between the proteins will change. So when the when the salt concentration increase, the binding strength becomes more strong.
Okay, thank you. Mm-hmm. And next question, Abdul raised a hand, so go ahead and ask your question.
Yeah, thanks for the talk. My question was the undulations that you showed, is there a dependence of the wavelength on the bending rigidity?
Okay. Yes. So I don't quite I'm not very clear with the simulation part because it is done by our collaborator. So yeah, but I think the general idea is we applied we applied a compressive stress to the to one lipid to the outer leaflet of the lipid bilayer and see what what is the energetically favorable result.
Thanks. So the next question in the chat is if you always see a single tubule formation, or if you can see several, or sometimes see several.
Yes. No, we sometimes there's only one, but I think that the shape of the tubule and the number of tubules varies in different vesicles. Yeah, in the same under same condition, we can see some tubules has a lot of tubules inside, but some of them only have one. So yeah, we can see both.
Thank you. There's another question, whether you, by Sagar, asking whether you ever have tubules that reach the other side of the membrane. And, also, if you know what sets the length of the tubules.
Oh, so yeah, in my experimental part, I didn't count the length of the tubule because it's very hard to see the full tubule. Only when the tubule is in the light in the same layer, in one layer, you can see the full length. But in many circumstances, we can't see the full length of the tubule. So it's hard to determine the length of the tubule.
Okay, a lot of questions. So Mason wants to ask a question, so go ahead.
Mm-hmm.
Yeah, this was awesome. In your multi-component membranes, was there any evidence to suggest that the tubules were enriched for a specific lipid component? Like, did the tubules have more POPC than the rest of the membrane?
Oh, so under our experimental conditions, there's all the temperature is above the melting temperature, so there shouldn't be lipid phase separations. So the lipid composition should be uniform in our vesicles.
Okay.
Mm-hmm.
So I think, yeah, ask one more question and then we move to the next talk. So the question is, what happens if you remove some of the excess area of the vesicle?
Remove some excess area.
Area, yeah.
That's a good question. What do you mean by remove some area of the vesicle? Sorry, yeah.
I don't know exactly. Maybe it's more okay. By sucking some of the vesicle, for example.
Sorry, can you say that again? Yeah.
But maybe using some device to suck away some part of the vesicle.
Oh, I don't quite. I'm not quite sure about this question. Yeah.
Okay.
I haven't think about change the area of the vesicle.
Okay, cool. Thank you very much. I think we should move to the next talk then.
Mm-hmm.
Yeah, thanks, Sam. So next talk is the Alexander Atimin from the Bulgarian Academy of Sciences. And the talk will be about the role of DIO2 in DNA replication and regulation of cell growth and division.
Okay. Can you hear me, everybody?
Yes, we can hear you.
Okay, so let's get right into it. I think you see me, right? So yes, my name is Alexander, and thank you for the introduction. Before I start, I would like to thank everybody for giving me the opportunity to talk at this forum and for listening to what I have to say and giving me the opportunity to present part of the work that we do at the Institute. So basically, we're going to talk about two proteins, DIA2 and CTF, involved in the replication in yeast. But before we talk about them, let's talk a little bit about what is replication. This is a process in which basically the DNA of every living organism doubles. And this is a process that is very complex and a lot of proteins are involved, like in all of the steps, starting from initiation, elongation, termination, regulation even. And without going into a lot of details in this complex model, we're going to show and work with this simplified model. Now, in this model, we have the CMG helicase, which is composed of the MCM complex, the GINS complex, and CDC45, which together act as a helicase which unwinds DNA and basically creates room for the other proteins to fit in. When this happens, there are the polymerases, which are a lot and are very different. And basically, their main function is to synthesize DNA. Now, another important players or key players in our model are the MRC1, CTF4, and DIA2. MRC1 is a protein that basically acts as a sensor, transmitting the signal that there is some problem ahead of the replicon, so the cell can react accordingly. And CTF4 is a protein that is involved in the stabilization of polymerase alpha, which is the main priming enzyme. You can say that this is the main protein in the beginning of replication. Now, when we talk about replication, we have two opposite replicons moving against each other, and at some point, eventually, they will meet. This will happen in the termination of the process of replication. Now, when this happens, here we have a little problem. If these two don't collide, there will be DNA in between them, which won't be accordingly synthesized and replicated. So people have found that actually in this model, DIA2 acts as a complex part of SCF ubiquitin ligase, ubiquitinates MCM7, which acts as a binding and recognition site for CDC48, which disassembles the MCM complex, and after that, disassembles the whole replisome. So the polymerases can finish this DNA in between the two replicons. Now, also, people have found that MRC1 gets degraded in a DIA2-dependent manner in the process of replication recovery. This is a process in which when there is a damage to DNA, the cells recognize this damage, they repair it, and after that, or some problem, it's not necessarily damage or some problem, the machineries, the cellular machineries recognize this, they repair it, and after that, the process can continue normally. And for this to happen, MRC1 needs to be degraded. Okay, with knowing all of these things, we asked ourselves, is DIA2 somehow involved in CTF4 metabolism? Because we know that DIA2 binds to the replisome thanks to MRC1 and CTF4. They act as its stabilizers to the replisome. So we know that DIA2 is involved in the degradation of MRC1. Is this the case of CTF4? So what we did, of course, logically, the first step was to delete DIA2, and the second step was to put the green fluorescent protein reporter at the CTF. First, we asked, are the cells okay? Are they viable when we do this? And as you can see from the viability assay, they feel perfectly well. Here we have also MRC1 strains acting as additional controls to see that everything is normal and the cells feel good in this environment. Now, when DIA2 gets deleted, there is decreased viability, especially when we add the agent, the inhibitory agent, hydroxyurea. This is an agent that blocks replication by diminishing the pool of nucleotides in the cell. And yes, the cells are viable, although they don't feel as healthy as the cells with intact DIA2. Now, the next step was to see what happens at a singular cell level. So we conducted a lot of live cell imaging experiments. And as you can see from the graph immediately, at the end of the G2M phase of the cell cycle, there is higher amount of protein in the strain where DIA2 is deleted. Now, we wanted to see if this is a single time event or this happens with the bulk of the cells. So we conducted whole protein extracts. And what we saw from them was that at the 30 to 40 minutes recovery time point, we have increased amount of CTF4 when DIA2 is missing. Now, in this experiment, first you synchronize the cells with hydroxyurea at the beginning of S phase, then you wash the inhibitory agent, and you see what happens. Now, this 30 to 40 minutes time point, we needed to see to what stage of the cell cycle it corresponds. So we conducted a FACS analysis. And as you can see from it, the cells are either with duplicating or duplicated DNA. So this means that they're in the G2M phase of the cell cycle of the yeast Saccharomyces cerevisiae. Now, after that, we wanted to see where is this protein located. So we conducted an assay in which you can distinguish between protein bound and free protein. And again, as you can see, most of the CTF protein at the same time points is onto the chromatin. Now, we did some digging and we proposed the following model. Now, on one hand, DIA2 is responsible for the ubiquitinylation of MCM complex. So when there is no DIA2, there is no ubiquitinylation of MCM complex. So there will be intact replisomes at the chromosome. There will be nothing to be disassembled, and the complex will stay bound onto the chromatin, and this will explain the higher levels of CTF4 onto the chromatin. On the other case, probably DIA2 is somehow involved in the metabolism of CTF4, and thus we observed its higher amount either in bulk cells or onto the chromatin. Now, something interesting else that I want to show you is that when we have a normal cell cycle of yeast cells, they look something like that. They divide between 120, 160 minutes is their normal cell cycle, they're round. Now, when you remove DIA2, and this is, I believe, very interesting, some of the cells start acting pretty crazily. First, they remain bound to each other, and also they start elongating. And what's most interesting, they multinucleate, and the nucleus starts going randomly in the cell, dividing at random points, moving around. So this phenotype becomes even more extreme when you add this hydroxyurea agent. When this happens, basically the cells stop looking like singular unicellular organisms, but more like a multicellular one. And the nucleus basically starts acting like a bridge in between the daughter cells. When we conducted different analysis, the thing that we saw was first that the duration of the cell cycle was strongly increased, like almost 2 times longer than the normal cells. Also, the ratio between the long and the short axis of each cell was increasing. And finally, the area of individual cell was strongly increased when this phenotype is observed. Now, we did some headbanging, basically brainstorming, and we know that the cell has 3 important checkpoints that basically should prevent what we're seeing. The first checkpoint is the DNA damage checkpoint, which basically detects if there is a DNA damage and stops the cells from progressing. On the other hand, in yeast cells, there is the spindle assembly checkpoint, which checks if every chromosome is bound to the spindle and if they can proceed to the next stage. And finally, we have the spindle positioning checkpoint, which is responsible for moving into the daughter cell, because yeast has a special type of chromosome segregation, and basically to separate the 2 set of chromosomes in each daughter cell. Now, as we can see first, we have a constant phosphorylation of RAD53, which is a very strong marker for persistent DNA damage. So obviously this checkpoint is not working because the cells are dividing. On the other hand, we see that the nucleus is not stopped before dividing, and the cell cycle is not controlled because the nucleus can divide at random points in random cells at a random place. So obviously these 3 checkpoints are not working properly in these cells, and somehow DIA2 is involved in all of this. But we don't know yet how. We need to conduct some extra research, and hopefully in a year or 2, I can answer any questions about this. So these are all the people that were involved in all of these experiments, and I would like to thank each and every one of them. And I would like to thank all of you for your attention. If you have any questions, I'm at your disposal.
Thank you very much, Alexander. Sure, there are questions?
Okay.
All right. Any student has a question, otherwise you can ask a question.
Yes.
How do you know that they're not alternative mechanisms? Basically, you did these things by deleting some stuff, but are you sure there's not another pathway for that, or you think it's just this particular pathway that you proposed?
Well, it is possible. We did a lot of, people have been working with these proteins a lot. And basically there are a lot of debating what actually happens with the CTF protein and what is its metabolism. And this is also a key protein in higher eukaryotes like ANT1. And they have proposed a lot of mechanisms for degradation or disassembly. But it seems most logical to be like this, because on the one hand, you have this mechanism. And as we know, living cells usually copy the same mechanism for partnering proteins. So if one goes in one pathway, it's logical to think like this. But of course, it's possible there are other pathways, first of degrading, of disassembling, or of control of these processes.
That's correct. I agree with you. It's a very, very nice work and appears to indicate that. But I always, when metabolism is involved, I get very nervous on what's cause and what's consequence. You know what I mean? You have a mechanism that affects metabolism, or metabolism is controlling the mechanism. So it's a very, sometimes cause and consequence becomes a problem. It was very nice work. I really enjoy it. Thank you.
Thank you. Thank you for the question.
Hi, could I ask a question?
Yes.
Do you know if DA2 somehow affects the helicase activity in terms of timing?
Yes, okay, that's a very good question. As far as I know, there's no direct effect of DA2 onto the helicase activity. I think it's only, it only recognizes it when it's bound. Still, I'm not completely sure how it exactly recognizes that this is a termination site and how it doesn't degrade it at earlier stages or disassembles at earlier stages. There are some theories that maybe when two replisomes meet, this is the signal for it to be recognized, ubiquitinated, and then degraded. But as far as I know, there's no direct effect of DA2 onto the helicase activity.
Thanks.
I think there was a question by Sane before. I'm not sure if that's still
answered in the response to Jose. Okay. Thank you, though.
Yeah.
I have a quick question, if it's still allowed.
Yes.
Yes. So we did look at DNA replication and cell growth in E. coli. And I know experimentally they could see that the cell started to grow for like about, it was a slow growing E. coli cell for about 20 minutes before it started to replicate the DNA. So it, it would be interesting how to put in the circuitry. So many people have started to look at particular pathways in the lipid metabolism that might be connected somehow to this system. Have you looked into bacterial systems in great detail?
No, we haven't. We haven't. At this point, we haven't. Perhaps in the future we could start expanding, but mostly we're focusing on yeast and higher eukaryotes.
Yeah,
at this point.
Yeast is like a bacteria.
Well, yeah, it's very interesting, yes, because it's fast dividing, it's easy to look after, it's not very pretentious about the conditions, and yeah, it gives good results. I agree with you.
Yeah, so I'll send you our work on it. All right, thank you.
Thank you, thank you.
Good. I think if there are no more questions, then we can move to the next talk. So thanks, Alexander, again. And the next talk will be by Zane Thornburg from University of Illinois, Urbana-Champaign. And he will talk about stochastic spatial simulation of gene expression in a minimal cell.
I need the Whoever's sharing needs to,
Oh,
there,
yes.
yeah. Thank you. Should see presenter mode now. So hi, everyone. My name is Zane Thornburg, and I'm a graduate student in Zan Lucy Schulten's group at the University of Illinois. And I'd like to start by thanking NSF, CPLC, and PoLS for giving me the great opportunity to present my recent research in simulating stochastic spatial simulations of gene expression in minimal cell day. So first of all, let's talk about the minimal cell. What is it? So we're working with a real minimal cell. This organism is named JCVI Syn3A. It's a synthetic bacteria that was synthesized by our collaborators at the JCVI, Clyde Hutchison and Hamilton Smith, with parent organism Mycoplasma mycoides. And as you can see by the gene maps on the left, we have a dramatically smaller genome than E. coli. So we only have 493 genes instead of 4,600. And in E. coli, 40% of the genes have unknown function, whereas in our genome, currently only 20%, as currently annotated, have unknown function, but that number is going to go down with a new annotation in the near future. So we have a good idea of all the processes that are going on in this cell. So today I'm going to talk to you about the genetic information processing reaction network. So we're going to simulate replicating the DNA as the last talk did, and then we're going to talk about transcribing DNA into RNAs, and then translating mRNAs into proteins and degrading mRNAs back into nucleotides. And so we simulate reaction networks in our group using something called the reaction diffusion master equation. So the reaction diffusion master equation discusses the time evolution of the probability to be in a state X, where the state X contains all coordinates and chemical identities for each particle in the system. So in the reaction diffusion master equation, we have a lattice-based model where you can diffuse from one sublattice to a neighboring sublattice. So in any time step, you can diffuse just to a neighboring lattice site, or you can react within one sublattice. And so the reactions, the first term, the reaction term, is known as the chemical master equation, which you solve using a well-stirred assumption. And then you just have kinetic parameters such as rate constants and whatever you want. And in the RDME, particles have to be in the same sublattice, but you can assume, if you assume your whole system is well stirred, then you can just solve the chemical master equation, which is what we do first to test our reactions. So to begin building our reaction network, we'll start with the initiation of DNA replication. So in bacteria, the initiator protein DNA-A is responsible for initiating DNA replication. And so it's been found through crystal structures that its domain 4 binds to double-stranded DNA to 9 base pair signatures, and then that its domain 3 binds to single-stranded DNA and 3 nucleotide repeats. And so using the sequence information for the genome of Syn3A, we have 2 low affinity sites and 1 high affinity site for the domain 4. And then through the mechanism, we have an AT-rich region next to it. So you'll bind your 3 DNA-A to the 9 base pair signatures. Those will cause some torsional strain on the DNA, which open a pocket of single-stranded DNA, to which domain 3 binds in the 3 nucleotide repeats. And using the 91 base pair AT-rich region, we should form a filament of length 30 DNA-A. And the binding of the DNA-A has been measured using SM-FRET, and we use those kinetic parameters in our model. Then for replication, transcription, and translation, we use a polymerization model. So here we have a mechanism where your enzyme will bind to a template polymer. So your enzyme is DNA polymerase, RNA polymerase, or a ribosome will bind to DNA, or in the case of translation, your ribosome binds to your mRNA. And then once you've bound, you go through association steps of associating monomers, so nucleotides or charged tRNA, and then you'll go through elongation steps of forming your chemical bonds in your polymer. And so in this rate form, the 2 most important factors are your elongation rate in nucleotides per second, amino acids per second, and then the dominating term in the denominator is the total length of your mRNA. The other terms become significant in cases where you're depleting your pools of your monomers. So if you're running out of nucleotides or charged tRNAs, then your polymerization rate will be depleted. And then in the case of a spatial simulation for the reaction diffusion master equation, we require RNA polymerase to diffuse to genes, and we require messengers to diffuse to ribosomes so that we can include the binding reactions separately. And so in our well-stored model, we can simulate 1,000 cells over the course of a whole cell cycle. And so in the inlaid graph here, we have 4 individual cells, the length of the DNA A filament over time. And you can see that to initiate DNA replication, it can take anywhere from under 5 minutes up to 40 minutes in this cell using the kinetic parameters we obtained. And then when you plot the distribution of all 1,000 cells, we found an average replication initiation time of 8 minutes. And then once you've initiated replication, our polymerization model predicts DNA replication to be completed after about 65 minutes. And so the only part that I hadn't mentioned yet was mRNA degradation. So in the well-stored model, in the CME, we assumed a uniform half-life of 4 minutes among all messengers. And so in the top left corner here, you can see the balance of, you can see mRNAs degrading or being formed stochastically over time. So this is the messenger count for the glucose 6-phosphate isomerase mRNA for a single cell over the course of a cell cycle. And at 60 minutes, you can see that the mRNA counts become higher due to gene duplication. So once you've duplicated your gene, you have higher mRNA counts because now there are 2 copies of the gene for your RNA polymerase to transcribe. And so we can take these, this data for every single one of the 1,000 cells for every of the 452 mRNAs and average them. And so we get plots like those on the right, where you can see that for metabolic proteins and ribosomal proteins are the ones I chose to show here, we average over 1,000 cells anywhere from less than 1 up to almost 7 messengers for any messenger in the cell. And then our experimental metric that we compare to, we have genome-wide proteomics for this cell. So over the course of a cell cycle, you'd imagine you'd like to double your number of proteins on average. And so here, the x-axis of the histogram shows you the scaled protein counts over after 105 minutes after a cell cycle. And these are all scaled for each protein to its own proteomics count. So we'd hope to get a histogram centered around 2 for doubling the number of proteins. And that's exactly what we obtained with just several outliers that are all either proteins with really small proteomics counts or are very short, so they're produced very quickly. So now we've got a reaction network that's working reasonably well. So now we need to build a spatial model. So we need some knowledge of cell geometry. So we have some cryo-electron tomograms from the lab of Elizabeth Villa at UCSD. And we like to use these to obtain ribosome coordinates and get knowledge about parameters and other large bodies in the cell. And so we have a distribution of wonderful tomograms of cells that are earlier in the cell cycle to ones that are late in the cell cycle to get an idea of what the ribosome distribution looks like. And also with cell geometry, we can also, in the spatial model, we can place complexes of interest if we have knowledge about them. And for this case, we're in a gram-positive bacteria. So we have a mechanism known as a complex known as the degradosome. And the degradosome in gram-positives is composed of 2 metabolic enzymes, enolase and PFK. And then it's made of 2 exoribonucleases, RNase R and RNase J, an endoribonuclease, RNase Y, and then a helicase. And so this is the mechanism that degrades mRNA. And in our simulations, we require, in the spatial simulations, we require the messengers to diffuse to these to be degraded instead of a uniform decay rate. So to build our cell simulation, we go from our tomogram, we obtain the ribosome coordinates and put them into the simulation. So we have yellow spheres as the ribosomes. And then my group mate, Benjamin Gilbert, wrote some code that places the DNA, and we have circular DNA. So the DNA here in the bottom picture is colored such that the red is the origin and then blue is the end of the DNA. So you can see that it does return to itself, and we have circular DNA that we've placed around our ribosomes. And then you can place degradasomes, which are the red spheres, and then you can place the 77,000 proteins from the proteomics. And so in this simulation, I was able to simulate this for 20 minutes, and we have, whenever a ribosome turns purple, that means it's actively translating a messenger. And whenever a red degradosome turns blue, that means it's actively degrading a messenger. And so you can see that we have diffusion. And if you pay attention to some of the ribosomes as they turn on, if they're close to a degradosome, once they turn off, you'll notice the degradosome turns on not long after the ribosome turns off. because the messenger is likely diffusing to that degradosome to be decayed not long after translation. And so these spatial simulations are more computationally expensive than the ones for the well-stirred assumption for the chemical master equation. So we averaged over 5 cells, and you'll notice from the video that we had the ribosomes fixed and did not duplicate the DNA. So we only go out to 20 minutes since we haven't programmed gene duplication in yet, but you'll see that the messenger counts on average for metabolic proteins are smaller, but instead of the uniform mRNA half-life, we obtain a distribution of half-lives, and we obtain an average half-life of 1 minute, which is in agreement with Mycoplasma galliseptica measurements, and then the ones at higher half-lives are in agreement with some Bacillus subtilis measurements. It's still a little faster than we'd like it to be, but this is a very, very good first result for this sort of simulation. So we just need to incorporate a few other parameters that I'll discuss in the future directions. And then to our metric of the proteomics counts. So since we're not simulating over a whole cell cycle, we obviously don't want to double the proteins yet. So after 20 minutes, we'd expect about 20% of each protein's proteomics count to be generated if you assume linear growth. And then, but as you can see that our distribution is not centered around 1.2 as we'd expect then. Instead of it being centered around 1.2 or producing 20%, it's centered a little lower than 1.1, and the average of this is 10% of the proteomics counts are being generated. So one thing I neglected to mention with the well-stirred model is that we accounted for polysomes in that model. And in this model, since the ribosomes are fixed, we cannot incorporate polysomes yet. So this is likely due to our lack of polysomes in this model compared to the well-stirred model, where polysome just means you can have multiple ribosomes actively translating a messenger at any time. And so in conclusion, we were able to simulate the reaction network of genetic information processes for the minimal cell and double the counts of most proteins over the course of a cell cycle for the CME. And then requiring the mRNA to diffuse to degradasomes definitely results in a distribution of half-lives and a more realistic estimate of mRNA degradation. And so work in progress, we are working on integrating our CME simulations with an ODE model of the metabolic reactions, and we'll later do the same with the RDME. I'm going to assign diffusion coefficients of each messenger and protein based on hydrodynamic radius instead of uniform diffusion coefficients. And then we're going to incorporate polysomes, gene duplication, and cell growth into the RDME. I'd like to thank my advisor, Zan, Elizabeth Villa for providing the tomograms, Benjamin Gilbert for writing the code for the DNA placement, and the programmers Julio Maya and John Stone, with whom I was able to modify our code so that it can even handle the size of this model. Thank you for listening as well.
Thank you, Zane. We already have a first question by Mauro, who is asking, so the location of the ribosomes that you took from the experiment, the first question is, are they randomly distributed? And then the follow-up is, what do you expect to happen if the ribosomes can move?
Okay, so the answer is I haven't done statistics yet on the, here we go. So I haven't done statistics yet on the actual ribosome distribution, but it's relatively random. The only thing that is somewhat consistent is you get a good number that are close to the membrane. You get a good number of ones that are close to the membrane. And then in terms of if they could diffuse, then once 2 ribosomes get close to each other, we could have them actually form a polysome. So you could have 2 ribosomes actively translating a messenger. Since they're fixed, they can't get close enough to each other to interact in most cases. So that'll be what we can get from that.
Thanks. Any other questions? Oh yeah, next question is by Shubham, who is asking, are there some genes whose expression gets doubled earlier as compared to other genes during DNA replication? And if so, does this have biological implications?
Yes. So the biological implications, I'm not sure. But so in gene duplication, so in bacteria, since you have circular, actually, this is a good illustration right here. So since you have circular DNA in bacteria, your origin, which is here at the border between the red and the blue, is where replication will start, and it will go in either direction. And that is the only location that it will start. So the genes that are closer to the origin on either side definitely get replicated earlier. But in terms of what that means for this cell, I'm not sure yet. We have done some studies in our group on E. coli that look at some other things that in fast-growing E. coli that show some importance, but I haven't looked at anything for this cell.
Okay. I think if there are no more questions, we thank you again, and it's time to move to the next talk, which will be by Abdul Maumey Kakare from UT Austin, and he will talk about Catherian clustering during vertebrate embryo elongation.
Hello, can you see my screen?
Yes, we can see and hear you.
Okay, sounds good. Yeah, so I'm Abdul. I work with Dr. Dave Tirumalai. I'm excited to tell you about this work, which is a collaboration with experimental developmental biologist, Dr. Robert Hubner from John Wallingford Lab. He was supposed to give this talk, but he fell ill, so I'm filling in for him. So understanding axis elongation is a fundamental question in developmental biology, where embryos, they go from, at initial stages, they resemble a ball of cells, which are roughly spherical in shape, and then they elongate along an axis in order to form the head to tail axis. So this is an important problem to understand. This is important to understand because the failure of the embryos to elongate appropriately can lead to many birth defects or congenital anomalies. So if you look at this graph here, the death rate in newborns per 100,000, the death rate in newborns due to congenital anomalies are much higher than that of Alzheimer's, stroke, diabetes, and flu put together in adults. So we want to understand how cell behaviors contribute to the elongation of the embryo. And I'll be focusing on this collective cell behavior called convergent extension. So, a simplified picture of the convergent extension behavior is given in the cartoon on the right here, where what is shown is a cartoon of 4 cells, and the junction between them is labeled. This is what we'll call the V junction. I'll call it the cell-cell interface. And what happens in convergent extension is that this cell 2 and 4, they come together by shortening the junction that was present between them. They come together to meet, and then they extend this contact, which is what we'll call the T junction. So you can imagine these cell behaviors, they contribute to elongation of a tissue. So the cells are initially distributed laterally this way, and then they converge in this axis in order to extend the tissue in this other axis, which is where the name convergent extension come from. So these are super resolution images that are obtained by our collaborator, zooming into these junctions. So you can see the cell outlines here and the vertices at the end of these junctions, that those are indicated by the arrows. And the junction length here is indicated by L of T here initially. So in this movie, this length initially is about 10 microns. So the terminology vertices and junction length is what I'll keep referring to again and again. So this is an important mode of collective cell behavior that's not well understood, especially the mechanical underpinning, and how the molecular underpinnings of this contribute to this cell movement. So prior work in terms of understanding cell movement, they have considered these single cell-cell interfaces to be mechanically homogeneous. They have assigned uniform mechanical properties to these cell-cell junctions. But our analysis of super resolution imaging and modeling tells us that we need to rethink this picture, that these single cell-cell junctions are really mechanically heterogeneous entities. And I will make clear what I mean by mechanics as we go on. So one of the initial clues that we got in terms of this asymmetry in the behavior of the vertices is that, so here is a movie again of vertices that are contributing to this shortening of the junction. And the same image is shown on the right as still pictures. What you can see is that the vertex that's labeled by this yellow arrow, it contributes much more to this shortening compared to the other vertex. So this is what we will refer to as an active vertex, which contributes much more to the shortening, and then the other one as the passive vertex. So we can quantify this by the distance moved by the vertices as a ratio of the initial junction length. And then when we look at these junctions across many different embryos, what we see is that there is clearly an active vertex that contributes dominantly to the shortening compared to the other vertex, which we will term as the passive vertex. So this type of asymmetric vertex dynamics was also recently reported in another system, a fly embryo. Now we look to quantify the dynamics of the vertices. So we calculated the mean square displacement. And what we observed, so this is red is for the active vertex and blue is for the passive vertex. So there are these three distinct regimes where at short times, both vertices show mean square displacement exponent of 1, indicating Brownian dynamics. And then at an intermediate time, the passive vertex dynamics slows down, showing subdiffusive dynamics with an exponent of 0.8. And then at a later time, both of them recover to show superdiffusive movement with higher superdiffusive exponent for the active vertex. So this intermediate slowdown in the dynamics of the passive vertex is an indication of glass-like behavior. We quantified this behavior further by using the self-overlap parameter. It tells you, the self-overlap parameter tells you the likelihood that the vertex will move over a distance, a specified distance LC as a function of time, with a value of 1 indicating that the vertices have not moved, and a value of 0 indicating that they have moved from their positions. So what we see is that the overlap parameter shows very distinct relaxation between active and passive vertices, with a double exponential behavior for the passive vertex and a single exponential behavior in the tail for the active vertex. We also quantified the dynamic heterogeneity. It tells you the variation in the relaxation of the overlap parameter, and we see this distinct difference in both the peak time as well as the peak height of the peak between the active and passive vertices. So we conclude from this that these active and passive vertices show distinct dynamics, and this is an important feature of this collective cell migration called convergent extension. So next we built a model in order to understand and predict how this behavior comes about. So in this model, what we think about, so these vertices are shown, active vertex in red blob and passive vertex in blue blob. And we think about the movement as being, of these vertices as being driven by a piston, which is then attached to a spring. Of this stiffness of the spring is given by KL, and then this stiffness of the spring is given by KR. And then one can write down the equations of motion for the position of the left and right vertices, depending on the spring, and then this force term coming from capturing the effect of this piston. So from the model, we see that a key time scale emerges that's given by the ratio of the stiffness of the spring to the friction term. And this is an inverse time scale. And this is what I will refer to as the viscoelastic ratio, which sets the time scale over which the vertices will relax stress. Now we explored the parameter space in the model. So here I show the parameter yield exponent. It tells us how quickly the piston is moving. I will not focus on that, but focus on the viscoelastic ratio. So here we are varying the viscoelastic ratio for the active vertex, where the passive vertex viscoelastic ratio is fixed at a small value. And what is shown in color is the blue regions indicate that there is a very low probability for the junctions to shorten effectively, while these yellow colors indicate that there's a high probability that these junctions will shorten effectively. So what we see from the theory is that when the viscoelastic ratio is symmetric for both the vertices, there's a high probability that the junctions will not shorten. While if the viscoelastic ratio is asymmetric and it's larger for the active vertex, we go to this regime of very high probability of junction shortening. So then we can recapitulate experimentally observed features of junction length behavior as a function of rescale time. So here, this is the change in junction length as a function of rescale time from both experiment and theory for junction length changes from many different embryos. What we see is that in this regime, the junction length behavior are highly self-similar across the embryos, and they are well described by this compressed exponential. While in the non-shortening regime, what we see is that the junction length change behavior shows both experiment and theory show large deviations away from this compressed exponential behavior. So we wanted to understand what could be the molecular underpinning, molecular reason why this difference in viscoelastic property near junctions could come about. And we predicted that what could happen is that near active vertices, you have these cell-cell adhesion molecules called C-cadherin, that they are clustered with a larger spatial clustering near active vertices, while near passive vertices, they show these smaller clustering behavior, as shown in this cartoon here. In order to explain cadherin, so this is a cell-cell adhesion molecule. Here is a single cadherin molecule, and this is a cartoon of 2 cell membranes. And these single cadherin molecules, they jut across the membrane and then link to the cadherin molecule from other cells in order to mediate cell-cell adhesion. So this is an individual cadherin transdimer. So many of these transdimers, they can cluster together, as shown here, 4, and then a larger number of transdimers showing larger and larger cluster sizes. So the mechanism of clustering is well studied, but then what function clustering plays, especially in terms of embryo development, is not known. And we also predict that there should be a difference in the local junction stiffness that should be dictated by the clustering of cadherin, as motivated from some of the earlier work. And as we came up with a way to quantify the cluster sizes of cadherin using the spatial correlation in the cadherin intensity fluctuations as a function of distance along the cell-cell junction. So you can see at short distances, they are highly correlated, and then this correlation decays as you go farther and farther away along the junction. And what we see is that this is averaged over many different, over 1,000 different instances in time from many different embryos. And what we see is that passive vertices show quicker decay in the correlation compared to the active vertices. While if we look at in the same embryos, non-shortening junctions, this difference in the correlation in cadherin clustering goes away. There is no palpable difference. Here we extracted the individual cluster sizes by looking at the distance over which this correlation goes to 0. And what we see is that active vertices show enhanced cluster sizes compared to passive vertices, while the passive vertex cluster sizes are similar to non-shortening cluster sizes. So in order to give you a summary, I've shown you that the movement of vertices during convergent extension, it displays asymmetric and glass-like behavior, and cadherin clustering is patterned locally with large clusters near active vertices. And both theory and experiment suggest that cadherin clustering is tuning the local mechanics, the viscoelastic property of the junction near it, in order to facilitate the shortening. And then I didn't have time to go through this, but dynamic clustering of cadherin is essential for these movements that we observe. And then I would like to thank my experimental collaborators here, and then our PI, Dave Thirumalai, and then the rest of the Thirumalai lab. Thank you.
Thank you very much, Abdul. Other questions? Maybe we allow faculty questions for now to start.
How you make your choice of parameters? Are they all phenomenological? They come from other experiments? They come from the same experiment? I got a bit confused how your choices come.
Choices for which?
For example, for your ratio, your force friction ratio, your elastic. I'm trying to figure out which the real physiological regime from your plot and how you get into these parameters.
Oh yeah, so for the viscoelastic ratio, we made sure that the ratio that we obtain are consistent with other reported values from fly embryos, for example. So we made sure of that. But then this K parameter here, we couldn't directly measure it, this entire parameter. But we came up with a way, if you look at our preprint, to measure the transverse fluctuation of the membrane and then link it to the stiffness.
Yeah, but the stiffness may depend on more things than just the membrane, right? The entire cell is involved. That's why. Yeah,
there could be other contributions, but then this work here from a group in France, they show that if you prevent these cadherin from clustering in this way, it changes the stiffness of the junction. So they showed this in vitro cells, and our experimental collaborators also made mutations of this to prevent this clustering, and we can show that this asymmetry goes away.
Okay.
Okay, so we have a few more questions in the chat. So the first question is there a reason why the correlation function does not decay to 0?
So the correlation function decays to 0 for both of them.
Okay. And then next question is, during neural tube formation, stem cells align in a circular fashion to form rosettes. Could your model include collective behaviors such like this?
Yeah, rosette-like behavior is another interesting contribution to how this neural tube forms. We have not looked into that, but that's something that we can look into in the future.
Okay, and then I think the last question by Ria is this vertex activity observed in all kinds of cells?
So in this particular case, we are focusing on mesenchymal cells in frog embryos, but this asymmetric vertex behavior was also reported recently in fly embryos. So we think that it's a conserved feature across different embryos.
Okay, let's also ask the last question that just appeared. Is there any role of myosin in this kind of cadherin clustering?
Yeah, so we looked into seeing whether actomyosin, if there is any differences in the actomyosin behavior next to active versus passive behavior, and we didn't find any difference. So we think that the cadherin clustering is what is transducing the actomyosin fluctuations to affect this movement of the vertices.
Okay, cool. Thank you very much, Abdul. And let's move to the final talk for this session by Joris Messelink from LMU in Munich, who will tell us about the distribution of single-cell chromosome conformations.
All right. Good afternoon, everybody. So today I will tell you about inferring the organization of the bacterial chromosome. Let's somehow, okay, this works as well. So the question that we're interested in is how ordered is the bacterial DNA? To give you some sense of the length scales involved, a typical bacterial cell is around 2 micrometers long, whereas the entire circular chromosome is around 1 millimeter. So this implies the chromosome has to be highly compacted to fit inside the cell. But at the same time, functions like transcription and segregation have to be regulated in some way. And so the classical idea was that there's actually not that much spatial organization going on. And the DNA can be thought of more or less as a random polymer sitting inside the confinement. However, with more ongoing experiments, more spatial features have been discovered. And now the picture is more of a structured polymer. And so the state-of-the-art method to investigate spatial chromosome organization is a so-called high-C map. And in this map, we see how often genomic regions come into spatial contact with each other. So I'll now show you the high-C map for Caulobacter crescentus, which is the one that we'll study today. And so this is what it looks like. And so along the axis, we have the genome positions, and the color tells you how often specific regions come into contact. And so we see a main diagonal. So these are regions that are genomically close, and we see they have a higher contact frequency, but we also see a lot of domain-like structures appearing. So it definitely behaves differently from just a random polymer. And secondly, we see a second diagonal appearing, which can be interpreted as the two arms of the circular chromosome being folded on top of each other. So just from looking at this map, we can already infer some features of spatial organization, but we would want to go one step further and extract a distribution of three-dimensional configurations from such a map. And the question is, can we do this just directly from the data without inferring any model of the underlying dynamics of the chromosome? And it turns out this is possible. and such a method of inference is known as maximum entropy inference. I'll now quickly explain to you what the idea of this method is. So the starting point of the method is the distribution of probabilities P of chromosome configurations R. And for each of these possible configurations, we demand that it fits inside the cellular confinement. And now we write down the entropy of this distribution, and we want to maximize it, but under a constraint, which is that the contact frequencies of the model have to match the contact frequencies from the experiment. And now if we do this constrained maximization, we obtain the equations for our probability distribution P of R. And so this is determined by the Lagrange multipliers that we introduced for each of our constraints. And we are now faced with an inverse problem of finding the values of these Lagrange multipliers. And we solve this using iterative Monte Carlo simulations. And I won't go into the details of that now, but I'll just show you the results. So here we see our contact frequency map again. And in the top triangle, we now have the experimental values. And in the bottom triangle, we have the model, which, as you see, matches the experimental contact frequencies very closely. So this tells us that we've managed to converge our model, and we, in fact, found the maximum entropy model for the bacterial chromosome. So now that we've done this, we can generate lots of single cell configurations, such as the one I'm showing here. And so just as a guide to the eye, I've labeled the origin in green and the terminus in yellow. And with our algorithm, we can now generate lots and lots of single cell chromosome configurations and then ask questions about spatial organization from them. So the first question that we asked is, can we challenge this maximum entropy model by independent experiments? And so I'll show you one of such experiments in which they fluorescently labeled 4 different genomic regions on the chromosome, which you see on the left. And then for each of these regions, you see the distribution of positions along the long cell axis. And if we were to have just a random polymer, the distribution would look like this for all regions. However, for the maximum entropy model, this is what they look like. And so we see that the two actually match quite closely. So this tells us that the model, in fact, has predictive power, and we can learn things from predictions of the model. So the next question is then, what novel biological features can we extract from the model? And to explore this question, we made use of 2-point correlations between genomic regions. So these tell you how the position of one specific genomic region depends on the position of another genomic region in the cell. And so the first one I'll show you is angular correlations. So imagine looking at a cell from the top, then this tells you what the relative positions along the main axis are of the regions. And we see that correlations decay quite rapidly away from the main diagonal. Now, we can do the same thing for the radial correlations. So how far are the regions away from the center? And again, the correlations decay very rapidly. And lastly, we can look at the correlations along the long cell axis. And here we find a striking pattern of correlations along the entire chromosome. So just to clarify what we're looking at here, if we would have one genomic region, say the one labeled in purple, and we look at how it moves along the long cell axis, and then look at another region further on and see how this one moves at the same time, then we find that there is an anti-correlation along the second diagonal. So basically, if this one would move to the right, this one would move to the left. Whereas for the main diagonal and the regions over here and here, we have positive correlations. So this we found quite an interesting result, and we dug a bit deeper. And we found that the answer to what causes these correlations is found in the single chromosome states, specifically in the distances between regions. So here I'm showing you a map of distances between genomic regions for one specific state. And as you see, there are clusters appearing. between regions that are close in space, and these are kind of separate from other clusters. And this turns out to be a ubiquitous phenomena. And what we can do is just mark these clusters with some algorithm that picks them out, and just label each of the domains that we find. And so each of these domains we termed a superdomain, mainly because it is so large. And the interesting feature is that if we would now take an average over all single cell states, the structure disappears again. So what we see from this is that the structures are quite variable, and they're not localized to specific points. And so I won't show you all the details of the analysis, but so what we basically found is that, yeah, they are quite large, they're variable and delocalized. And we found what the connection is to this correlation map, namely that domains from opposing chromosomal arms have a tendency to avoid each other. And so this means if we have a group of domains from one chromosomal arm and the other group from the other arm that overlay, then as soon as one tends to move to the left a bit, the other would move to the right to keep on avoiding it. And this induces the anti-correlation pattern that we see. Now, lastly, we also considered the information contained within the position of chromosomal regions. So imagine we would have some protein or protein cluster diffusing through the cell and then attaching to a region on the chromosome. This would give it some information about where it is in the cell. And with this model, we can quantify how much information can actually be gained from this. And so if we would have a random polymer, the localization information would be a bit less than 1 bit. Whereas for our maximum entropy model, we find that this information reaches up to 3 bits for the origin and terminus. And so this tells us, on the one hand, that there is quite a bit of information contained in here that could be used by the cell to guide processes. But on the other hand, there is also still quite a lot of variability. So we can't really think of the chromosome structure as one rigid object either. So now I'll just give you a quick summary of what we've seen today. So we developed a maximum entropy model of bacterial chromosome organization, which gave us the distribution of single cell chromosome configurations. We revealed pronounced large-scale features that can be explained by mutually excluding superdomains. And we quantified the amount of positional information that's stored in chromosome configurations. And with that, I'd like to thank you for your attention, and I'll take any questions.
Thank you very much, Joris. Are there student questions? Yes, so we have first student question by Ben, who is asking if you can make any statements about whether a chromosome configuration is not.
That's actually an interesting point. So one thing that we're limited by is just the resolution of the high C data. These are regions of about 10 kb. And our model tells us how subsequent 10 kb regions are arranged in space. And on the substructure in there, we couldn't see. But of course, you could also look at larger scale knots that might appear in the chromosome. And this we haven't really considered yet. And it would be an interesting question to look at.
Next question is by Antonio, who is asking what methods you use to compare experimental and simulated high C maps.
So to compare the maps, one thing we did was just look pairwise, or make a pairwise comparison of all the pairs of regions. And then you would expect, if you plot the contact frequency of the experiment and the model, that they would all lie exactly along a straight line. And so we looked at this and we calculated the Pearson's correlation coefficient. And this was, well, like 0.999 more or less, which indicates that they are very close to each other.
Thank you. Next question is by Arvin. Do you have mechanical properties between euchromatin and heterochromatin in your model?
I'm not completely sure. So what kind of mechanical property should I think of? Maybe Agavit can elaborate a bit more.
I guess the question is if you have substructures depending on which DNA binding proteins might bind and modify the structure of the chromosome.
Yeah, so one thing we did look at is the high C maps of two mutants, where, for example, one mutant where they knocked out SMC, which is a protein that loops around the chromosome to organize it. And we indeed found that looking at this knockouts, we have quite different pattern of large-scale organization. So basically the correlations become much stronger and the properties of these superdomains also change a bit.
Okay, a lot of questions. So Shubham asks whether you think that the anticorrelation between domains on opposite arms is simply a physical phenomenon, or does this have any biological significance?
Yeah, I mean, this is a good question. So yeah, basically, does the cell, can it actually do something with these correlations or not? We're not really sure if the cell is actually making use of this. But we do find that this could be a mechanism for long-range coordination of the chromosome. So in principle, there is information there that the cell could use. Whether it actually does, we're not sure yet.
And then Sumit Daba asks whether you constrained the positions of the Ori and Ter regions, or if they're allowed to diffuse.
They are allowed to diffuse. So the only thing we did to get our localizations is to just break the symmetry of the cell by defining one half of the cell as the origin half, because otherwise you would always have a symmetry around the middle of the cell. But apart from this way of breaking this left-right symmetry, they're allowed to freely diffuse around.
Okay, then Andre asks why you fixed the sum of the frequency times the Lagrange multipliers to 0.
Yeah, good point. So this actually, maybe I can quickly go back to this so everyone sees what this one is about. So yeah, it's the bottom equation here. And this one actually arises from the fact that the contact frequency map is normalized. We cannot really make a comparison between absolute contact frequencies between model and experiments, which basically gives us a free parameter for the overall scale of the contacts. And the way that we dealt with this was by treating it as a parameter in a maximum entropy model and then maximizing the model entropy under this overall scale of the contact frequencies. And then if you demand that the entropy is maximized, you get this as an additional constraint on your energies. And this indirectly fixes the scale of the contact frequency map.
So I think there's still quite some open questions. So maybe you can answer them afterwards in the chat. But I think for a matter of time, thank you again for your talk. And I want to thank all the speakers and also everybody who was asking questions and contributing to a lively session. And yeah, so I think now it's time for poster sessions, and I want to remind you that there are more talks tomorrow. I think, Margie, maybe you want to say something about the procedure now.
I have resent everybody the links to the speaker chat room and to the poster viewing rooms, so you should have that available. Tomorrow we will start as usual at 10 o'clock Eastern time. We have 5 talks, and afterwards we'll have a discussion about planning the meeting that we mentioned in the email on Wednesday or Thursday night or Tuesday night, Wednesday morning. I think that's it. I think that's it.
Okay. Yeah, and thanks everybody again. And time for poster sessions then.
Yes, thanks all. See you at the posters. And thanks, Kurt, for excellent moderation.
You're welcome.
And excellent talks, everybody. Very enjoyable. Really impressive.
Dan, can we stay there for a little bit? I just need to talk to you.
Sure. Hang on, let me kick a kid out. Amelia, can you go in the other room? No. Wrong answer. Please go in the other room. No. Okay, then just hold your ears. Yeah, okay. So we're going to
Hi, Margaret. I'm waiting to talk to Dan. I won't be able because I had other obligations tomorrow to be on the meeting. Can you help Dan on organize the discussion at the end of the meeting? Can I give you that responsibility? We have to get some.
I don't feel that I'm going to lead this discussion on.
I think we just have to get a few volunteers.
Yeah, I'm going to let the leadership do this. This is outside my Amelia, please go away. I need to talk to them now.
Okay, I'll talk to.
Yeah, I'm going to let, this is outside my purview. This is a national thing. I am not the right guy. Okay. I don't also have the bandwidth right now. So I'm going to punt on this one. And I apologize.
All right. Bye-bye then.
All right. Thanks.
Hello. Tomorrow,